All right. I don't know why my camera looks a little distorted today. I think I'm on the wrong camera. I'm on this one up here. All right. So let's talk a little bit about energy. We're continuing Unit 3 from yesterday for AP Biology. So we just reviewed those laws of thermodynamics that say energy cannot be created or destroyed. Energy is only moved around, and things become more disorganized over time. At this point in your scientific careers, everybody knows what that triangle thing, though, that equation's all about, right? What are those? Hang on, I got there. What are these triangles right here? Delta. What does that mean? Change. Okay, that means change. So the change in free energy or available energy in a system it determines how likely a reaction is going to happen. Okay, so listen, we could set a bar of steel in this room and leave it here for 1,000 years, and it would slowly rust. But there are things we could do to make that more favorable or less favorable. Okay, so free energy is going to determine how likely a reaction happens. Okay, and this equation's pretty easy. Don't get too intimidated by the equation. It's mainly about looking at changes in a system. Okay. Moving on. I'm going to move this thingy out of the way. All right. So, like I said, free energy determines whether something's going to happen spontaneously. Okay? So that kind of means like on its own. That means no energy is required. So that no outside energy is required. It just the chemical just breaks down, or the chemical bonds, okay, on its own. There are ways, though, to make that more likely. And so what we do, we look at how free energy changes, and then we classify reactions as exergonic or endergonic. You have probably heard words really similar to this called exothermic and endothermic. Those are subcategories of exergonic and endergonic, okay? Gonic, or I should say ergonic, means work, and erg is Greek for work. It used to be a unit in physics, and erg was a unit of force. That was in the sort of the imperial system. Don't ask me how big it is. I don't know. Look it up on Google what an erg is, okay? So exer sounds like exit, so it's sort of like exit work. Ender means within, okay? So there's some sort of work or energy within. So even though it means work, we're talking about energy, okay? So energy is something that's really hard to define when you really get into physics. Some of the brightest physicists in the world, Feynman he said, we don't even know what energy is. It's weird. So exergonic reactions, remember, exit energy, they are reactions that release energy. An example of this is cellular respiration. When cells take in glucose and oxygen, they release energy in the form of ATP, okay? All this means is less energy is needed to break the bonds than there are in the products that are released, okay? So it takes a little bit of energy to get it going, but then more energy comes out. Less energy is required to get it started than what comes out. I think of this as maybe like a catapult. Okay, here's my little catapult. I'm going to bend back my rock thrower. and I probably have like a little string or something here or a lever, and when I put a little energy into that lever, the whole thing goes off. Another way you could look at it is a whole bunch of dominoes. Those are supposed to be dominoes. You barely push the first one and it releases all the energy in them. So you have, what kind of energy do you have stored up in the catapult and the dominoes? Potential is exactly right. So we are focusing on chemical potential energy here and it's called Gibbs. Well, the energy that's released is what we're talking about. The chemical potential energy that is released is what we're talking about. When reactions are exergonic, energy is released from the products. So you're looking at products right here, and they have a lower amount of energy. If you follow the laws of thermodynamics, that energy couldn't get destroyed. It went somewhere to do something. So the energy didn't like disappear. It's just no longer in the system of whatever reaction this is. So there's probably some heat given off. There's probably some motion. There's probably some cellular transport. Something happened, and now the energy of the products is lower. This is usually ATP turning into ADP. Okay? Let's look at the opposite side of this. Endergonic. That means within energy. So what we have here are things that are taking energy in to break or make bonds. And what we have is we start with a lower amount of energy and we end with energy stored in that system. And the products, so compare from here to here, you can kind of see the arrow that measures that. Energy has to be put in from outside of the system. Outside the system might be the sun, and photosynthesis is endergonic. Okay, endergonic. We don't really think of photosynthesis in terms of heat. That's why we don't use the words like endo and exothermic. Okay? We say gonic. Okay. Everybody okay? Pacing okay? Everybody seems to be keeping up with me over there. So let's look at this on a cellular level. I've been trying to relate it to cells the whole time because this is a biology class focused on cells. When cells are alive, they constantly have things going in and out of the membrane. Cells are not at an equilibrium state with the environment. Why is that? What if cells didn't take in any more energy? They would eventually do what? According to the laws of thermodynamics, the cell would become so what it couldn't live anymore. The cell stopped taking in energy and material. What law of thermodynamics would say it would shut down? You don't have to give me the number, just give me the term. If nothing could come in or out of the cell, what's entropy going to do inside the cell? Increase or decrease over time? Cells can become more disorganized over time. Things will break down and the cell can't maintain homeostasis. So, because cells are not equilibrium, they have to do work to stay alive. And that means the energy is going to be taken from the environment, material is going to be taken from the environment, and the cell is going to use that material to do three kinds of work. First one is mechanical. That's what my mouth is doing right now, speaking, breathing, okay? My pen moving around. All right, tons of that mechanical work I'm doing require transport. My nerves are pumping sodium and potassium back and forth across membrane. My muscles are also doing something very similar with actin and myosin, okay? So transport is pushing things across the membrane. And the other type of work that cells do is chemical. They are making new molecules, okay? Right now, very, very slowly, your hair is growing, your fingernails are growing. Just in this class period, you've probably had millions of red blood cells wear out and your body needs to make new ones. Okay? If you wonder why you're tired all the time, it's because your cells have to do all this stuff. All right? Take care of your cells and they'll take good care of you. So we have three big types of work: mechanical, transport, and chemical. Now there are some animals that do all kinds of weird, funky work, like make sounds, which is mechanical, but you have things like lightning bugs that glow, that's really cool, different kinds of work, okay? You have things that can like camouflage themselves. They do that by transporting things in and out of cells that are different colors. Really cool stuff. All right, how's this all work? I'm going to go get my little model, and I'll be right back. So we have this molecule. I better not talk so they can hear me over there. All right, now I'll talk that I'm back close to my microphone. We have this special molecule called ATP, and I like to use this baby toy as a model. This is my adenosine, okay? And this is my three phosphates, these three guys, red guys right here. These phosphates are very, very high energy molecules, and they sort of repel each other, and they have a lot of stored energy in them. So knowing that, what ATP does is it joins exergonic reactions to endergonic reactions. So exergonic activities make endergonic things happen, okay? What does that mean? Well, when this thing breaks down, we have ATP goes to ADP. And hopefully you're looking at my little picture here where I'm taking the three things and breaking them off. So this one phosphate group breaks off. That's going to be exergonic, okay? Energy is going to exit this process. It's breaking down. It's catabolic, okay? And so this breaks down, and that turns ATP triphosphate into diphosphate, okay? So here's what we have. Organisms get energy that they need to do those three things we talked about by breaking this molecule. There's some organisms that can also even break ATP or ADP down where you have a single phosphate when they're really low on energy. We're not going to get into that. We call this process, breaking this phosphate off for it to do work, we call it, it's a big word, phosphorylation, okay? So this released phosphate is going to go give some other molecule energy, whether it's a protein pump, whether it's actin or myosin, whether it's a motor protein that's pulling something through the cell. It all happens the same way, phosphorylation. Now, when we phosphorylate, what would you guess we use to break the bond? What kind of molecule do we use to break the bonds in sugars? And say it loud. That's right. So we call this, what do we do when water cuts? Hydrolysis. That's right. This is the hydrolysis of ATP. So water comes in and breaks this thing off, and this phosphate phosphorylizes something, gives it the energy, and then usually this gets recycled back into ATP. I hope you can see me on the little camera. I feel like it's not working that great today. All right. So you can see the water moving there. You see the little yellow explosion. My special effects budget is really low, but that's sort of what we're looking for right here. The water moves in, breaks the bond, and what we get is ATP turning into ADP. Okay, so we get a phosphate that breaks off and energy, as you see down there in the little cartoon. Now, where does the energy come from? Okay, this is complex, but it comes from lowering the free energy. Okay, when this is held together, it really, really wants to break apart. Now, after it breaks apart, the free energy is lower. And like I said, we regenerate ATP by what's called the ATP cycle. What organelle does this happen in mainly? Starts with a mito, ends with a chondria. Mitochondria, wow, you guys are good. Okay, so what we have here is a cycle that goes over and over again. One side of it is exergonic. The other side of it is endergonic, or used for endergonic processes. All right. What can we do to control how fast this happens? Let's take a look. The rate. So thermodynamics will tell us they tell us if something will break down, but they don't say anything about how fast it will break down. So you can probably imagine that biological reactions need to be fast, not slow, okay? If something's chasing Mr. Sater out in the wilderness, I need to burn that energy quickly, not spontaneously. Well, I'll just wait for that ATP to break down. No, I need it to break down right now, okay? So the rate is what we're really concerned about. So some reactions are almost on a geologic time scale. So they are so slow, if we just let them happen on their own, that cells really wouldn't be able to use them efficiently. So if I take sucrose, table sugar, and just let it sit, it takes about 1,000 years for it to break down into glucose. So if I just let it sit in a beaker, put it in a vault, 1,000 years later, it will spontaneously break down. But what I want you to think about really quickly is cells don't have that kind of time. I don't know about you, but I can barely wait for my YouTube ads to skip. So I don't have 1,000 years, all right? So just think for a second. Cells can't wait that long. What do they do to speed up the 1,000 years? There's a substance in cells that lowers that energy threshold. What is it? Enzymes is exactly right, okay? So normally I put you into groups to do this, to talk it over, but we're not doing that in this day and age. So if it takes 1,000 years, how's it gonna do it? The answer, somebody said over there, is enzymes. Okay. Enzymes lower the amount of energy it takes for reactions to happen, and they happen way more quicker. They improve that rate. Okay. So let's take a look. What exactly is an enzyme? Okay. An enzyme is a macromolecule that speed up reactions by lowering the activation energy. All right. I work with a lot of plastic, plexiglass, and wood at home that's really thin. Sometimes I need it to break. If I just spontaneously bend it and it breaks, it never breaks on the straight line I want it to break on. But if I take a carpet knife or a utility knife and I just scratch it right in a line and then break it, it breaks exactly where I need it to. It's easier to break. That's kind of what an enzyme is, okay? I take that knife and I scratch down the side of the plastic and it'll break right where I need it to. That's kind of what an enzyme does, all right? So it takes a lot less energy for me to break that because I score it or get the break started. I don't know if it's a perfect analogy, but I do it a lot, so that's how I think about it. The cool thing about enzymes, just like my carpet knife, I could use it over and over and over again, okay? Enzymes are a type of protein. So you have a DNA, RNA code for all the different enzymes your body needs. And when you need more of a certain enzyme, there are signal pathways that will allow your body to react and produce these enzymes that are actually needed. Now, most enzymes end in ACE. Most enzymes end in ACE. Okay? So, you might call my enzyme that breaks down my plastic utility knife ACE. Okay? I know it's not real, but I'm just giving you an example. There's an enzyme called DNA helicase. There's an enzyme called ATP synthase. All right? So, what do we call the plastic? Okay, the enzyme is the carpet knife, but the thing the enzyme acts upon, it's called the substrate. It's what the enzyme acts on. Okay, the enzyme is going to act on the substrate. So if you have an enzyme that breaks down some sugar, that sugar is the substrate. Now, I have a carpet knife, and the only part of it that touches the plastic is the blade. So the knife has a big handle, it has a thing to pull the blade in and out. The place where all the activity is happening on my knife, that's the important part. And we call that part of the enzyme the active site. Okay? So whatever the substrate is, it goes into the active site. So a lot of the enzyme isn't really about connecting to the substrate. A lot of it's just sort of what I'm going to call molecular machinery. Pacing okay? Good. All right, so let's talk about how they do this. Enzymes have what we call an active open site that substrates can bond with, okay? The substrates are held together briefly by weak interactions. When I cut my plastic, I just hold it with my hand. So I kind of hold it in place for a minute. Then I need to let go of it and do something else with it, okay? The substrates, while they're in the enzyme, are then converted to products. And that might be building or breaking, okay? Enzymes can do both catabolic and anabolic things. In this case, it looks like we start off with two separate substrates that are changed or modified into this new yellow and blue configuration here. All right. This is kind of neat here. Once the substrate plugs into the enzyme, it will change its shape. Okay? Once the enzyme has the substrate plugged in, it will change its shape. It almost looks like a hand grabbing something, doesn't it? Like from the side, like a thumb and finger. All right. So I said this already, but let's break it down into steps. We call that induced fit. Enzymes can catabolize, catabolic reactions. What did I say the cat did to the chair? Tore it up. So our little pet cat with lots of energy tears things up. So when we have a catabolic enzyme, it's going to break things down. So this little blue arrow thing goes into the purple enzyme. It's going to hold it in place, and it's going to do something on a molecular level to make it easier to break with lower amounts of energy. Without the enzyme, this might only happen at a really high temperature. And it does basically, it gets pretty complex, but it puts strain on molecular forces and makes it easier to happen. All these substances in the enzyme really, really work on the substrate. The next one is anabolic enzymes. They help build things up. They help build things up. All right, looks like everybody's getting caught up over there. So I'm going to go ahead and stop this right here, and we will talk about the second part of this later. And that will be like, what are the effects on enzymes? And if you put enzymes in non-Goldilocks conditions, it kind of affects how they work. All right, we'll end this lecture here today.